Well, I guess I should have included this at the end of the last video, but we'll just start this one with it instead. This is a review of the endomembrane system, and including in that the synthesis or making, modification or changing, and shipping do something with these proteins. So the rough endoplasmic reticulum synthesizes or makes the protein and then puts it into a transport vesicle. Now guys, you can see the steps are numbered in the picture and you've got the words. Easy peasy, right? So we put the protein in a vesicle and it moves from the rough ER into the Golgi apparatus, where the vesicle fuses with the membrane of the Golgi apparatus and the contents are emptied into the Golgi. Now, those proteins are going to be modified as they continue to move through the Golgi apparatus. And once they are done, whether it's adding a sugar to the protein or a lipid to the protein, or chopping off part of the protein or hooking one protein to another, whatever it is, the modified protein is then going to be packaged up in another vesicle and released from the Golgi apparatus, where it will then go out to do whatever job that protein was created for. So these secretory vesicles will either merge with the cell membrane and insert molecules into the membrane, as you see up here, and this is becoming part of the membrane now. It also may release the contents by the vesicle fusing with the membrane, and then the contents are released into the interstitial fluid. So that's exocytosis. Or the vesicle can also take these enzymes. Remember, proteins are enzymes are proteins. So if it contains enzymes that are needed by the lysosome, this vesicle will fuse with this vesicle and form the lysosome. Now, our next organelle that we're going to talk about is also a membrane-bound organelle. However, it is not a part of the endomembrane system, so we consider it separately from that. Mitochondria are sort of bean-shaped. They're sort of oblong-shaped organelles that have a double membrane with the folds of the inner membrane being called cristae. Oh, this should be sounding familiar to you, huh? Of course, because the mitochondria are involved in cellular respiration. Remember, making ATP. The inner part of the mitochondria is called the matrix, and it has little circular fragments of DNA that's used for producing proteins that are used by the mitochondria. So they contain their own DNA. They help us to make ATP. So for this reason, they are called the powerhouse of the cell. Now, I know you've all heard that before. I could hear you saying it as soon as this slide came up. So remember, mitochondria make ATP in the process of cellular respiration. They contain their own DNA, RNA, ribosomes, and they can self-replicate. So the number of mitochondria in a cell increases by self-replication when there's greater demand for ATP. Ribosomes are organelles that contain proteins and RNA, and they're arranged in two different parts, or subunits, a large subunit and a small subunit. These are made in the nucleolus, which is located in the nucleus, but they're assembled or put together like puzzle pieces in the cytoplasm. Now, as we just talked about in the previous video, ribosomes that are attached to the endoplasmic reticulum form the rough ER. The rough ER synthesizes proteins that are destined to be exported out of the cell or to be put into the cell membrane or to be enzymes within those lysosomes. The free ribosomes are just sort of floating around in the cytosol. They make, in general, all the other proteins in the cell. All right, so this is one centrosome that is made up of two centrioles. The primary function of the centrosome is organizing microtubules that help to form the cytoskeleton of the cell. The centrosome, though, is best known for one job, and that is during cell division to form create microtubules that form spindle fibers. Those spindle fibers are then going to help to move chromosomes within the cell during that cell division. Well, if you look at this word, it should tell you a little something. Proteosome, protein body. All right, so these are little structures that are protein digesting organelles. So the proteins that they digest include those that have been damaged, or maybe they didn't fold correctly, or normal proteins that we just don't need anymore. So you can think of proteosomes as sort of the quality assurance control for making sure that the proteins that our cell creates and exports are proteins that are as they should be and usable and made properly. The cytoskeleton is a framework of proteins that supports the cell, just like your skeleton supports your body, but it also helps to organize the organelles within the cell. We have 3 different types of protein molecules that form the cytoskeleton. We'll look at each one of those just briefly. So microfilaments, micro should tell you small, these are the smallest components of the cytoskeleton. They form an interlacing web or network of proteins that helps to make that internal structure. It also helps to form cytoplasmic extensions called microvilli. We'll talk about those shortly. Microfilaments are also going to be located throughout the cell to help movement of organelles, vesicles, and other molecules within the cell, and they're also important in muscle contraction. Microtubules are the largest component of the cytoskeleton. They're arranged sort of like railroad tracks for directing the movement of organelles and vesicles within the cell. They also extend into cilia and flagella, which are projections off the cell. And let's see, that's all we're going to do on microtubules. The intermediate filaments, intermediate means between, so they're sort of in between the size of the microtubules and the microfilaments. They're less flexible than we have with the microtubules and microfilaments, and they function sort of like rigid rods to support the cell and to stabilize junctions between adjoining cells. Their protein differs in composition depending on where the cells are located in the body and what job they need to do. For instance, keratin is an intermediate filament that we find in the skin, the hair, and the nails. But we also have another type of intermediate filament that forms neurofilaments within nerve cells. So we just mentioned cilia, flagella, and microvilli. So let's talk just a little bit about those. So cilia are very small, hair-like projections that extend from the exposed surface of some cells. They contain those microtubules. So they line portions of the respiratory passage. Now, have you ever sort of felt that tickle in your throat and you had to cough? Well, sometimes that's because our cilia that line our respiratory passages are moving and propelling mucus back up toward our pharynx. You heard me again, pharynx, not pharynx. That's what we commonly call the throat, your pharynx. So the cilia move things along a passageway. Now, if you swallow or if you are eating and you get a tiny little piece of food down into your respiratory passages are lined with mucus, which is like a glue trap. So whatever it is gets trapped in the mucus, but we don't want it to stay there. So all these little finger-like projections of the cilia are moving because it's in a wet environment, sort of like seaweed moving in water. And that constant moving of the cilia propels whatever you swallowed, along with the mucus that it is embedded in, back up to the pharynx and you swallow it. So cilia are used to move things along a surface. Flagella are very much like cilia, except they're longer and wider, and there's usually only one rather than thousands that are associated with a cell. Now, the flagellum is used to move a cell, and the only example of a flagellum in the human body is the tail of the sperm cell. So the movement of cilia and flagella occur through the microtubules that are a part of their structure. The flagellum, of course, propels the sperm cell so it can swim, if you will. And the image you have on the slide is showing you microvilli. Part of the word is chopped off up here, guys. My apologies. That should say microvilli. Here you can see the microfilaments extending up into the microvilli. So they're thin microscopic extensions of the cell membrane. They're short and more narrow than cilia, densely packed together. They are supported by the microfilaments. Microvilli have a more extensive cytoplasmic membrane surface. So this gives more area, more surface area, I should say, for more efficient membrane transport. Now, just like not all cells have cilia or flagella, not all cells have microvilli. But we do find a lot of cells with microvilli throughout the small intestine, where that increased surface area allows for more nutrient absorption. So just imagine if this was a solid piece up here, so a solid cell membrane. The only place that we could absorb nutrients from our food products as it passes across these cells would be right there on the surface. But when we have these finger-like extensions, then those food products and the nutrients can be absorbed all along those little projections of the microvilli. So that increased surface area allows for more efficient absorption of nutrients. Membrane junctions are composed of integral and peripheral proteins that connect and support cells and provide an arrangement between cells within tissues to coordinate interactions. There are three main types of membrane junctions or cell junctions. The first we'll examine is the tight junction, where membrane proteins form strands or rows of proteins, and they're positioned sort of like spot wells, very near the free or apical surface. So you can see up here there's nothing attached at the top, so that is the free surface, also called the apical surface. So tight junctions will seal off the intracellular space, the space between the cells, and this prevents substances from passing unregulated between these epithelial cells. Tight junctions associated with epithelial cells that form the linings of the small intestine prevent digestive enzymes inside the intestine from moving between cells and damaging other body structures. You also find these in the urinary bladder to prevent urine from leaking through the bladder wall. I don't remember if I said the word or said the two words spot weld on tight junctions. If I did, disregard it, because the desmosomes form the spot welds. So desmosomes have a thickened structure called a protein plaque that's on the internal surface of the cell membrane. Numerous protein filaments are going to extend from the protein plaque through the cell membrane of the neighboring cells and anchor them to each other, forming a spot weld that holds the cells very tightly together. This arrangement provides a lot of structural integrity to cells that undergo a lot of stress, such as your skin. I mean, we're constantly moving and pulling. Think about every time you bend your elbow or your knee, you're stretching the skin that covers those structures. So the desmosomes keep the skin from separating and tearing. We also find these playing a very important role in cardiac muscle. From the time your heart is formed inside your mother's uterus until the day you die, your heart contracts the muscle approximately 75 times a minute. So those sheets of cardiac muscle cells are all pulling against each other as the muscle contracts. So the desmosomes are the junctions that prevent separation. Your tight junctions prevent leaking. Desmosomes prevent pulling apart. And our third junction between cells is the gap junction, which is made up of 6 proteins that are integral proteins. Remember, that means they go all the way through the membrane, and they're arranged in a little circular structure called a connexon. That connexon forms a small gap between adjacent cells, hence the name gap junction. This provides a direct way for things to move from neighboring cell to neighboring cell. Things like ions, glucose, amino acids, and other small solutes can pass directly from one cell to another through those pores. This is very important, again, in cardiac muscle, because cardiac muscle all needs to contract at the same time in a synchronous manner to move that blood through the heart and then out of the heart. So all of those cells need to do something called depolarize at the same time in the same area, okay? So those gap junctions allow sodium ions to move from neighboring cell into the neighboring cells, and that is going to depolarize those cells so that they can contract. Now don't panic, we're going to talk about all that later on a little more. And here you have your concept overview. This one is on page 134 and 135 of your textbook. It basically just walks you through the organelles and the cell membrane and their functions. Now we do still have a little bit to go. We've not talked about the nucleus, but that's where we're headed next.